So this is going to be our last lecture about this chapter, that is about cell. So we will discuss about cell membrane and its transport in this chapter. So if we look about the chapter introduction, then it is not like a simple topic because it is really important. So that's why I made a separate lecture specifically for this topic. So if we talk about yield analysis, then as you can see that this topic is like really high yield, and the topics that are related to it are medium yield. That means all the things that we are studying today are actually really important, and there are really high chances that MCQs are going to be appearing from these topics. So without wasting time, let's start our lecture. And first of all, what are the important topics that you need to know? That these are the high yield concepts that are really important. Then take a screenshot of this and then we will move forward. First of all, what kind of questions could be asked from this chapter? They can ask you about what is the mechanism and how ATP is used in transport, and does ATP is required in a specific reaction or not, because in IMAT 2024 they tested about membrane structure and how transport occurred, and in 2025 they talked about osmosis and roles of membrane proteins in different concepts. So that's why we have to cover every single concept that is related to transport and cell membrane in this topic. So first of all, the first thing that you need to know is about the cell membrane. So the cell membrane actually has two types of models that explain the structure of cell membrane. The first model is called fluid mosaic model and the other model is called unit membrane model. We will talk about both of these models, and first of all we talk about the unit membrane model. The unit membrane model is like less accepted in our society, and scientists don't use this model to explain cell membrane. So why are we still studying it if it is not accepted? The reason is because the unit membrane model tells you by exclusion what cell membrane cannot be. If we talk about cell membrane, then we have seen that cell membrane contains about three types of molecules in it. It contains first of all proteins, and then the next thing that it contains is lipid, and then it contains carbs. So proteins, when we did an experiment, we got to know that the protein makes up about 60 to 80%. Like these values are different in different books, so that's why I am using 60 to 80%. It can fall in between. Some books might say 60, 70 and 80, and so that's why I am not writing an exact value, because it can be different in Pearson and it can be different in Cambridge or your other high school books. That's why a range has been given, so you know that what the range could be, what could the answer could be. It could be between 60 to 80, and the lipid is 20 to 40% of the cell membrane. And if we talk about carbs, then carbs contain like minute amount, or you can say really low amount in our cell membrane. So as you can see that these are the three molecules that are making up the cell membranes, that is protein, lipids and carbohydrates. So if we know that protein, lipid and carbohydrates are making the cell membrane, then we can guess the structure of cell membrane using these three molecules. So the first thing that they thought about is that like maybe if protein is 60 to 80%, like it is so much in high amount that it should be the most occurring, or the one that should be in the most abundance in the cell membrane. So they said that the protein should have been on the outer ends like this. This is the protein, and then the lipid would be embedded inside the protein molecules. That's why it is also called sandwich membrane model, because the two outside proteins are sandwiching the lipid inside. Like there are two breads and then you have like salad and egg and chicken in between, like you are making a sandwich. That's how the protein is sandwiching lipid in between. That's why it is also called sandwich membrane model. That's what everyone thought, that the cell membrane would have a model like this, but this model was actually wrong. Why it was wrong? Because the thing was that the proteins and lipids were actually not in following these kind of structure. When they did experiments, when they saw different reactions, they thought about like this is not the structure that how it should be, because how would different type of molecules would move from this structure. Like if we have to pass, it would be really difficult to move from between the proteins. It would be really difficult for lipids to give the stability using this kind of structure. This kind of structure does not give the stability that we get in our real plasma membrane. So that was the questions that weren't answered by this kind of model. So that's why there was another model that was introduced, and that model was called fluid mosaic model. And fluid mosaic model is the most accepted model that is right now to explain all the aspects of plasma membrane. So this is the model that we'll discuss in detail in our chapter, that is about fluid mosaic model. Fluid mosaic model was given by two famous scientists. Their name was Singer and Nicholson. They gave this model in 1972. And there are two words that are used in this model, that is fluid and mosaic. Fluid is something that can flow. You have already discussed it in detail in physics chapter. Anything that can flow is a fluid. And mosaic means that anything that is like in a like varying or different or variable patterns. Like randomly, if you put something on the ground, that it is in a mosaic manner, right? So that's how you can say that the protein molecules are in a mosaic manner embedded in the lipid membrane. Just like as you have seen, you can see that there is a C. Let me make the C for you, like here. Imagine this is a big C, and then you have really big icebergs that are embedded in this C. So the C here is, what is the C and what is iceberg? The icebergs are actually protein molecules. Let's not write them here. The icebergs are what? The icebergs are actually protein molecules. And what about the C? The C is actually the lipid molecules. So this was the structure that was given by Singer and Nicholson, and Singer and Nicholson said that there is a sea of lipid, and there are really big icebergs that are embedded in that sea of lipids, and those icebergs are actually protein, and they are not embedded in a specific sequence. They are randomly embedded in the sea of the lipid, like there is no specific sequence. That's why this model is called fluid mosaic model, and the protein molecules are randomly embedded in the lipid. If you are thinking about like if the sea has more area and icebergs have smaller area, but if you see the diagram that I have drawn in front of you, that diagram is telling you that the sea is less. Like if you can see the diagram in front of you, then you can see that the sea here is small and the icebergs are really big. So that's how the actual structure of cell membrane looks like. The protein molecules are really large, while the two layers of lipid that are the C are really small in quantity as compared to the proteins. You don't have to literally take the example of C and icebergs, because obviously C is more than icebergs, but in this sense the lipid is C and proteins are icebergs that are embedded. So if we talk about the diagram here, as you can see, there are two types of proteins that you can see here. The first are the proteins that are embedded in the lipid cell membrane, like this. They are embedded in the lipids of cell membrane, and there are some proteins that are outside, like this. They are not intrinsic, they are not inside the whole cell membrane, they are only outside embedded, and they are called peripheral proteins. So there are integral proteins and then there are peripheral proteins. You can also say that these are intrinsic protein and they are extrinsic protein. That is the name. You can use any name that you want to, both are correct. You can say these are intrinsic and they are extrinsic protein. So this was all about the protein part of the cell membrane. And then as you can see, there are lipid molecules. They have two layers. So this is a lipid bilayer. It is not a single lipid layer but bilayer. Bilayer means there are two layers of lipid. The one is above, this is one layer, and this is another layer. So there are two layers of lipid, as you can see in the diagram. That's why it is called lipid bilayer. So that was all about the proteins and lipids. What about carbohydrates? Carbohydrates are not simply present, they are present in the form of conjugate molecules. Conjugate molecules means they are either with lipids or with proteins. They could be like glycoproteins, or either they could be combined with lipid and they could be glycolipid. Simple carbohydrates are not present. As you can see, the carbohydrate here is connected with a protein molecule, and as a whole it is called as glycoprotein. That means carbohydrate is not individually present in cell membrane. Either it is attached with lipid or either it is attached with protein, forming glycoproteins and glycolipids. So this was all about the proteins, carbohydrates and lipids basic. And let's go into detail for these molecules and let's discuss about the role of these molecules. The first is about the phospholipids. As you can see, the phospholipid actually forms the lipid bilayer. What does the phospholipids do? They forms lipid bilayer. This is the first molecule that is one of the most important one in cell membrane. So first, the other concepts that you need to know about cell membrane is that you can say it is cell membrane, plasma membrane, differential permeable membrane. There are so many names. I would be using different kind of names for cell membrane, so don't be really confused or don't think it is weird, because cell membrane has like 9 to 10 names. Okay, there are 9 to 10 ways you can call cell membrane, so I will use these kind of different names so that you will memorize these kind of names during the lecture. So I use cell membrane, plasma membrane, phospholipid bilayer. So we will be using these names in the whole lecture. The first thing is that phospholipid forms lipid bilayer of the plasma membrane. So the other thing that you need to know is that the plasma membrane is asymmetrical. What? Plasma membrane is asymmetrical, like there is no symmetry in our plasma membrane. Asymmetrical. The next thing that you need to know is that the proteins can drift sideways, and the thing is that proteins can also drift sideways. Proteins can also drift or move sideways. Like they can not move up and below, but proteins can move from left to right like this. They can either move left or right in sideways, but they cannot move above and below, and they also determine the most of the functions in membrane. So they can drift sideways and determine most of the functions, right, of our membrane. So that was all about the proteins and plasma membrane. The other thing that you need to know about that the plasma membrane is actually fluid. Like we talked about that lipid is a kind of a sea. It is a fluid, it moves like fluid. So that means cell membrane has fluidity. So how does the fluidity change? If we talk about the fluidity of our cell membrane or plasma membrane, then it depends upon some conditions. Like fluidity is not constant, it can change. So what are the things that it depends upon? So the first thing that you need to know is that it depends upon obviously it will depend upon lipid, because lipid is the C, right? The next thing that you need to know about fluidity is that it increases with the increase in unsaturated fatty acid. Like fluidity is directly proportional to unsaturated fatty acid. I have written it in the short form, so you can write it in your notes in complete, because it didn't have the space here. So fluidity depends upon the unsaturated fatty acid. Like the more unsaturated the fatty acid is, the more fluid it will be, because it will have double bonds in the lipid membrane, and if there are double bonds, then the fluidity would be really easy, because double bonds will support the membrane and the membrane would be more fluid than it is saturated. So let's move on to our next molecule, that is about cholesterols. So cholesterols actually stabilizes your protein. So stability of your cell membrane is from cholesterol. It stabilizes the membrane. And the other thing is that it also restricts the movement of ions and polar molecules. The other function of cholesterol is that it restricts, what does it restricts? Movement of ions and polar molecules. Like if you want to cross ions and polar molecules, they will be stopped by the cholesterol. Cholesterol will not let ions and polar molecules into from the cell membrane really easily, because ions and polar molecules can react with the cell membrane. That's why they are really dangerous, and they there is a specific pathway for ions and polar molecules to pass through the cell membrane. They cannot like come in the front of the door and pass through the cell membrane. Cholesterol is like, stop man, don't come close to us, because you can react with the cell membrane and you can damage it. That's why cholesterol restricts the movement of ions and polar molecules. The other about types of proteins. So as we talked about, there are two types of proteins, intrinsic and extrinsic. First of all, let's talk about intrinsic proteins. There are three types of intrinsic proteins. They could be channel proteins, they could be carrier proteins, or they could be pumps. So channel proteins are like allow a molecule or ion. What does channel proteins do? They allow movement of like a particular molecule or ion. It does not allow to like every particle to pass, but it mostly lets allow the molecules or ions to pass through it. That's why it is called channel protein. It is the channel of motion. If we talk about carrier proteins, then carrier proteins are like really selective. Like they does not allow most of the molecules to go through the cell membrane. They are really selective, and they only allow specific molecules inside the cell membrane. So that's why carrier proteins are called carriers, because they carry only specific things that they want to carry. They don't carry anyone that comes in front of them. If we talk about pumps, then pumps actually causes active transport. What is active transport? We will discuss it in detail in our end of that lecture. But active transport actually causes the motion of the things that we need in the cell. For example, if you have glucose and the glucose is present in the cell, but you still need more glucose in the cell, then what will it do? That active transport will cause to motion of glucose from low concentration towards high concentration. Like there is glucose in the cell, but you still need to get more glucose, then it will come into the cell through active transport. You don't have to be like really confused right now. We will discuss it in detail. So this is the like general idea I'm giving you. We will discuss every single molecule in detail. The next thing is about enzymatic action. So there is in specific enzyme in the proteins. Like some membrane proteins have enzyme functions. They perform metabolic reactions. For example, if we have example of adenylate cyclase, it is an enzyme. It actually catalyzes the transformation of ATP to cyclic adenosine monophosphate. Like adenosine monophosphate is converted into cyclic adenosine monophosphate by using this enzyme, and it catalyzes this reaction, and it also helps as a secondary messenger. Like for example, if a message comes to the cell membrane, then it will receive the message and it will become a secondary messenger, and it will take the message to inside the cell where the message is needed. So these are the specific enzymes that are also present in your cell membrane. If we talk about receptors, then there some proteins also acts as receptors, and they receive specific signals from other cells, and these receptors proteins are actually extrinsic. So receptors are the proteins that are extrinsic. This is the work of extrinsic proteins that actually act as receptors. If we talk about glycoproteins and glycolipids, they are also called as cell surface markers. Cell surface markers. Just like you mark something that you have. For example, if you have a pen, you will use an ink or something like a paper, or you will scratch the pen and say that this is my pen, this is my mark on it. So that's how glycoprotein and glycolipid are specific cell surface markers, and they mark the cell to show that this is the cell that have a specific meaning. And they also help in the recognition, like they also recognizes anything that comes into the cell, so they also helps in the recognition of anything that comes into the cell. So these cells are cell surface markers, and they also helps in the recognition in the cell. So these were the specific molecules that we'll discuss in detail now. The first thing that we'll discuss in detail is about the phospholipid bilayer. So first thing that you need to know about phospholipid bilayer is that we have already discussed this molecule in biological molecules. You know that we have discussed it. It contained two parts. The first was hydrophilic head, and then it had hydrophobic tail, right? If you remember, this was the head, and then you had this was the tail, right? First of all, let's talk about the head portion. So the head portion, as we know that it was actually polar. The head was polar and it was hydrophilic. Why the head was polar? Because of which molecule? Yes, because of phosphate molecule, right? Because of phosphate, the head was polar and it was hydrophilic in nature. Hydro means water and philic means loving. It is water loving. If we talk about tail, then the tail is actually non-polar. Tail is what? It is non-polar, and it is also hydrophobic. It is water hated. It does not like water at all. That's why it is called hydrophobic. It hates water. It has a phobia of water. And what is it is made up of? It is actually containing unsaturated fatty acids, right? This is the leg that is turned here. As you can see, this is showing a double bond that is here. If it has multiple turnovers like this, that it means it contains multiple double bonds. So it contains unsaturated fatty acid, and because of fatty acid it is non-polar, or you can say it is hydrophobic. It is hydrophobic because of fatty acids. It is also, you can say, it is thin and flexible. So this was about the phospholipid that we discussed in our biological molecule chapter, that it contains your three types of molecules. It contains glucose, it contains it contains carbohydrate, it contains fatty acid, and it contains phosphate groups. It is polar because of phosphate groups, and it is non-polar because of fatty acids. So if you can see that hydrophilic heads are actually outward towards the aqueous environment. If you see the plasma membrane, in the plasma membrane, this is how hydrophilic head is outside and the tails are inside, just like this. You can see the diagram here. So the heads are facing outward and the lipids are inside, and then in these lipids you have different kind of proteins and carbohydrates that are inside of these molecules like this, right? So the hydrophilic heads are in outward and the hydrophobic tails are from the inward. The arrangement is like thermodynamically stable, like no energy input is required for bilayer formation, and it is the most stable structure that you can see. Like as I have told you before, it is like a sandwich, and the hydrophilic heads are on the outside and hydrophobic tails are on the inside. So that's how you can say that the head is connected with the water and tail is saved from the water because it is on the inside. So that was about the phospholipid molecule. Let's move on to our next molecule, that is about cholesterol. As we discussed before, cholesterol is actually the regulator or fluidity regulator. It stabilizes the membrane, and it is in two type of conditions. Either it can be at high temperature or either it could be at low temperature. At high temperature, cholesterol actually restricts the movement of phospholipid. It prevents the membrane from becoming too loose or too flexible, and the structure would be lost, right? If something is too flexible, then it will not have any structure. And at low temperature, cholesterol will disrupt the phospholipid packing, and the cholesterol will solidify and make the phospholipid really hard. So at high temperature, the fluidity will increase. At low temperature, the fluidity will decrease. So you need to know that cholesterol increases fluidity at low temperature and decreases fluidity at high temperature. So that was about cholesterol, and that is why it is also called as the fluidity regulator. Next thing is about the proteins. We already discussed about the proteins. Let's go through the overview again. There are integral proteins. They are also called transmembrane proteins. They are present in the entire lipid bilayer. All the lipid bilayer contains integral proteins. They helps in transport. Why do they helps in transport? We discussed it before, because they are from top to bottom connecting the outside and the inside of the cell, while the extrinsic proteins were only present on the outside. They were not connected from the inside. That's why extrinsic proteins are used as a receptor, and integral are used for the transport. If we talk about peripheral proteins, they are mostly for support, signal transduction and enzymes. If we talk about channel proteins, carrier proteins, receptors protein, we have already discussed them in detail. Channel proteins allow specific ions, while carrier proteins bind specific molecules and change conformation to transport across. If we talk about receptor proteins, they bind hormones, neurotransmitters and different kind of molecules that comes in contact with the cell. If we talk about glycoproteins, as we have already discussed, they are cell surface markers, and they also helps in the cell recognition. If we talk about enzymes, we talked about adenylate cyclase. That enzyme, what does it do? That it converts ATP into cyclic adenosine monophosphate. You don't have to be really overwhelmed by this lecture. You can always take it again and again to understand these concepts and these definitions. So, or you can just go through the lecture notes from that will be given to you again and again, because these concepts are really important. Believe me, you don't need to miss these concepts in your IMAT exam. Next thing that you need to know about plasma membrane is the membrane proteins that we discussed. So the first proteins that are written here are integral or intrinsic proteins, and then you have peripheral or extrinsic protein. They are in front of you. The diagram is showing them in a really great manner. Then they could be monotopic, polytopic, and the polytopic could be alpha or beta. These concepts are not really important, but you need to know what is monotopic and polytopic. Monotopic contains only one chain, while polytopic contains multiple chains. And polytopic could be alpha helix or beta helix, beta plate, beta barrel. So these are the structures of intrinsic and extrinsic protein. So you can visualize the whole structure in your mind. Don't just memorize what is integral and what is extrinsic. You need to actually visualize the diagrams and make the whole video in your mind, what is happening in a cell membrane, how things are happening, how does it look like. When you see the visuals and everything in your mind, then it is really high chance that you won't be getting these MCQs wrong in your IMAT exam. Next is about the transport in the cell membrane. There are two types of transport. One is passive and then is active. First we will discuss about the passive transport. So the passive transport is the transport that occurs passively. Passively means it does not need ATP to do the motion. So it actually moves from high to low concentration. For example, if you have two jugs here like this, one jug and another jug, and they are connected with each other like this, and if you fill these jugs, if this jug is filled to the top and this jug is filled like this, then what will happen? The water will start moving from this jug to this jug. This is jug B and this is jug A. So the water will move from jug B to jug A. Why is that? Because jug B has more water and jug B has less water. So you don't need to take the water and just manually add the water from B to A. No, the water will go automatically from B to A. You don't need to do any work. So that's how passive transport works. In passive transport, the molecules automatically move from high concentration to low concentration. The first example of passive transport is simple diffusion. Like simple diffusion requires no membrane proteins, they requires no energies, they can move. Simple diffusion is for really small non-polar and lipid soluble molecules. So if the molecule is small, if it is not polar, like if it is a polar molecule, if it has a charge, then it will not go through the diffusion process, because it will be really hard to move that molecule, because polar or charged molecule can react with the cell membrane, right? So that's why it must be non-polar, and it should be lipid soluble. If it is lipid soluble, then it can pass through the lipid bilayer really easily, right? You know, like if it is lipid soluble, if it's just like saying that water cannot move from water. Like if you have water, then obviously water can move from water. So it is really easy that it should be small, it should be non-polar, and it should be lipid soluble. So the molecules that cross by simple diffusion are oxygen, carbon dioxide, nitrogen, steroid molecules, ethanol, urea, and small molecular drugs. What is the driving force? Like we have already discussed it, like from high to low it will move automatically, and the rate is affected by the membrane thickness. If we talk about the thickness of the membrane, you need to know that it is 7 nanometer. Like what is the thickness of the membrane? Let me write it down for you. The most accepted way is that it is 7 nanometer. That is the thickness of the cell membrane. That is in the most important books, and it is most widely accepted value. It can vary in higher books, but it is mostly accepted in high school books. 7 nanometer is mostly accepted. What is about osmosis? Osmosis is simply diffusion, type of diffusion, you can say. Diffusion could be for any molecule, but if the diffusion is specifically for water, then the diffusion is called osmosis. So osmosis is the diffusion of water from the membrane, that is selectively permeable membrane. Why is it called selectively permeable membrane? Because cell membrane only allows specific molecules to pass through. It does not allow everyone or anyone to pass through the cell membrane. That's why it is called selectively permeable membrane, or you can also call it semi-permeable membrane. So cell membrane, plasma membrane, selectively permeable membrane, and semi-permeable membrane, all of it these are all the names for the cell membrane. So next thing is that water will always move from high concentration to low concentration, or you can say that low solute concentration to a region of high solute concentration. What am I talking about? For example, if there is a place where water is high, then the salt should be low, and if there is a place where water is low, and then the salt should be high, right? This is the concept that will be explained in our next lectures, and that's how balance would be made. If at a place there is higher salt, then what will happen? If we take that this is jug A, and if we say that this is jug B, we are just taking an examples, right? So as you can see here, that the water is high and salt is low in this situation, and in this situation the water is low and salt is high. What will happen? Water will start moving from jug A to jug B. And what about salt? And the salt will start moving from jug B to jug A. So that's how you know that there are two things. Water is the solvent and salt is the solute. So water will move from high concentration to low concentration. Water is the solvent, so solvent will move from high concentration to low concentration. And if we talk about salt, then salt will also move from high concentration to low concentration. But the definition written here is that water will move from low solute concentration to high solute concentration. As you can see here, in jug A, the solute is low, the salt is low. That's why water is moving from low solute concentration to high solute concentration to balance out everything between A and B. When both A and B have equal amount of water and salt, then the process will stop, then water motion will also stop. So that's how osmosis occurs. Osmosis occurs between two places in which there is a difference of water and there is a difference of solute or salt. So the water can move through the lipid bilayer by simple diffusion, but in many cells it also passes through specific channel proteins, and these channel proteins are called aquaporins, and they increase the rate of water, like water will move really fast through these aquaporins. So that was about osmosis. If we talk about other thing, that is facilitated diffusion. Facilitated diffusion is diffusion that occurs through two types of proteins. One is channel protein and the other is carrier protein that we discussed before. So diffusion, facility diffusion uses this protein to transport polar or charged molecules that usually or not easily pass through the cell membrane. So what is facilitate? Facilitate means helping someone or something to go from one place to another. It is like a VIP pass through of the different molecules. Like charged molecules or polar molecules cannot pass through the cell membrane, so they will go through a VIP pathway. That pathway is what? Facilitated diffusion. So these polar or charged molecules cannot go through normal pathways, so they will take a VIP pathway, that is facilitated diffusion. And facilitated diffusion is a type of passive transport, and it also does not require any energy, and it moves substances from high to low concentration. So there are two proteins. First is channel protein, the other is carrier protein. So channel protein form permanent hydrophilic pores, while carrier proteins bind their specific molecules. So these are the two types that you need to know. That first is channel protein and the other is carrier protein. You can see the examples here. The examples are sodium channel, potassium channel, chloride channel, aquaporins. And for carrier proteins it is glue transporters, that is for glucose molecules, amino acid transporter, and they are like slower than the channel proteins, but they are more specific, right? Carrier proteins are more specific. They are really specific and really selective for the things that pass through carrier proteins. If we talk about difference between diffusion and facilitated diffusion, so if we talk about what does it requires, simple diffusion, as we talked before, does not require any proteins. Like the molecules can pass through simply through the lipid bilayer. If we talk about facilitated diffusion, then yes, they require proteins. And what are the proteins? They are intrinsic protein. Which intrinsic proteins? Channel proteins and carrier proteins. So what about the direction? They both have same direction. They will move, both of them move from high to low concentration. Is the saturation possible? No, because simple diffusion can pass through from any place in the whole membrane, while facilitated diffusion can be saturated because there are limited proteins, right? There are limited channel protein, there are limited carrier proteins, so it can be saturated. Like if there are like 100 glucose molecules that have to pass, but you can have only 5 to 10 channel proteins or carrier proteins that can help pass glucose through those carrier proteins. So that's why facilitated diffusion can be saturated, because it is limited. If we talk about specificity, simple diffusion is not specific, anything can pass through if it is small and non-polar, while facilitated diffusion is highly specific. Like carrier proteins only carry glucose molecule, amino acids, and it is really specific about its function, while simple diffusion is not specific at all. Like it can, if it is small, it is non-polar and it is lipid soluble, then it can just pass through. It could be any molecule. If it is small and non-polar, then it can have then it can use the pathway of simple diffusion. If we talk about examples, oxygen, carbon dioxide, steroids, ethanol, urea and alcohol, these are examples of simple diffusion. If we talk about facilitated diffusion, it contains glucose, that is a large molecule, amino acids are large molecules, and ions like sodium, potassium and water. Water is a polar molecule, that is why it goes through facilitated diffusion. If you can see here, this is the diagram for passive transport and active transport. So this is the diagram for facilitated diffusion. As you can see, it does not require any protein, and it just simply pass through the lipid bilayer. If we see facilitated diffusion, it is passing through the channel proteins and carrier proteins. So these were the examples for passive transport. Let's talk about active transport. So active transport is actually the motion against the gradient. What is gradient? Gradient is the difference, just like there is a difference of potential in physics. Like, you know, there is a potential difference. At one place there are high positive charges, and the other place there is a high number of negative charges. Then the charges will flow from high concentration to low concentration. Same is for active transport. The motion is against the gradient. What does it mean? For example, if you have 100 glucose molecules inside the cell and you have 15 glucose molecules outside the cell, shouldn't the glucose molecules go from 100 to 50, right? But the cell is really selfish. It does not let the glucose molecules go outside the cell. What it does is that it actually takes the glucose molecules that are outside the cell and take them inside the cell, because cell is really selfish, it is really greedy, and it wants more glucose molecules. It is an example, okay? So what does it do? It will take those 50 molecules inside the cell too, and as you can see, it is against the concentration gradient. It should be have gone from high to low, but it is actually going from low to high. That's why it is called what is it called? It is called active transport because it goes against the concentration gradient. So it was about active transport. Let's talk about what are the examples of active transport. The first example is about sodium and potassium pump. Sodium and potassium pump is actually present in your nerve impulses, right? We will discuss it in detail in your brain and development chapter. So first thing that you need to know about is that this pump causes sodium and potassium ions to move in between the nerve cell. So 3 sodium ions are pumped out and 2 potassium ions are pumped into the cell per cycle. It consumes 1 ATP molecule per cycle. So the direction is both ions move against their concentration. Because sodium ion is moving out, that means sodium is more outside. If potassium is moving in, then that means potassium is more into the cell. If we talk about net charge effect, then net loss of 1 positive charge per cycle occurs because negative interior and there is a shift of charges. Why is there a shift of charges? We will discuss it in detail in our nerve impulse lecture. So don't be really confused about this topic. These are the things that you need to just memorize. Don't go in too deep right now in this chapter. We will go deep into this concept when we are discussing about nerve impulse, synapses, and how cell these kind of impulses move in your nerve cells. If we talk about the diagram here, as you can see, the ATP molecule is moving from, this is low concentration to high concentration. So if sodium ions are inside, if you can see, if these are the sodium ions and these are also the sodium ions, so the direction of motion should be like from high to low. But in active transport it is actually opposite. The sodium ions will move from low concentration to high concentration. The same is for potassium ions. The potassium ions will move from low concentration to high concentration. Like these are the low concentration and these are high concentration. So inside and outside are also potassium ions. So this was example of active transport. Another example of active transport is secondary active transport. Secondary active transport actually exploit the sodium gradient. Like it is created by a sodium potassium pump to drive the uptake of other molecules such as glucose and amino acids, and sodium ions flows back into the cell and down their gradient. So this energy is used to pull glucose molecules against the gradient without directly using ATP. Like for example, you used active transport for sodium and potassium, and from sodium and potassium you used reverse active transport on a few sodium ions. For example, let me give you an example. If few sodium ions were moving inside the cell, right, and you just did a reverse of it, in this reaction ATP was used. If you can see, ATP was used, but if we reverse the reaction, then ATP would be released, right? And this ATP could be used for the active transport of glucose molecule, and that's how you can move glucose molecule into the cell without making another ATP molecule. You will not need ATP from outside. You can just create unlimited ATP using these sodium ions. You can like recreate ATP again and again, but this is not broken or cheat code, but you know that the ATP actually came from outside. This ATP came from outside, and then you just reused this ATP and you reused this ATP to move glucose molecule. So this is secondary active transport. This was primary. Sodium and potassium ions were using primary active transport, and glucose molecule used secondary active transport. So this is not broken. The ATP molecules are not unlimited using this process, but you will actually need at least one exterior ATP, and that one ATP could be used again and again to move that glucose molecule. If you don't understand this concept, then you can watch the lecture again and again to understand this topic. If we talk about other transport, that is about vesicular transport. Vesicular transport is actually motion of molecules in bulk, like large amount of molecules are moving inside or outside of the cell. It is called vesicular transport. There are two types of transport. The one is endocytosis and the other is exocytosis. Endocytosis means going inside and exo means going outside. It is not really difficult. You can see in the diagram here. The endocytosis has two types. The one is phagocytosis, the other is pinocytosis, and then you have receptor mediated endocytosis. Phagocytosis means the intake of solid molecules, and pinocytosis means the intake of liquid molecules. And if you talk about receptor mediated endocytosis, then it is for specific molecules that will bind to the cell membrane on the cell surface, and then they will trigger the formation of vesicles. The vesicles will form around the molecules. For example, if it is a solid molecule, it will come in contact with the cell membrane, it will form a vesicle around like this. If it is a liquid molecule, it will also form a vesicle like this. But if it is a specific molecule, then it will release specific molecules, and these specific molecules will come and attach to the receptors of the cell membrane, and the cell membranes will form specific coated vesicle for that specific molecule, and this is called receptor mediated endocytosis. Receptor mediated endocytosis is actually really rare. Phagocytosis and pinocytosis is the most common one. If we talk about exocytosis, it is the reverse of endocytosis. It is the process, for example, the vesicles from Golgi apparatus or secretis pathway that we discussed in our very first lecture. The vesicle will be removed from the cell by exocytosis. So this was about endocytosis and exocytosis that happens in the cell membrane. The last topic for cell membrane is osmosis and tonicity. We discussed about osmosis. It is actually the motion of water molecules from high concentration to low concentration, and tonicity is actually the process that describes the solute concentration of solution compared to the cell and outside environment. If we talk about tonicity, then there are three types of tonicities. The first is isotonic, that is the equilibrium stage, or you can say the balanced state. Then you have hypotonic, and then you have hypertonic. Hypo means less and hyper means more or higher. So hypotonic condition is the condition in which solute is less. You only need to know about the solute part, and then you can just do the reverse for solvent. So you need to know that hypotonic means that the solute is less in the cell, and for hypotonic you need to know that the solute is higher in amount or higher in concentration in the cell than the outside of the cell. For example, if you talk about hypertonic condition, the hypertonic condition is referring to the cell, not about the external environment. If you say that if the cell, let me show the diagram. If the cell contains hypotonic condition, then that means inside the cell there are less number of salt. That's why it is hypotonic in the cell. What about the water? Water would be, that is in the outside. The outside will have hypotonic condition, and it would contain higher amount of salt as compared to the cell. So if there is no reference in the question, for example, if they are simply saying that the cell is in hypotonic condition, then that means they are talking about the condition inside the cell. And if they're specifically saying to you that the water is in hypertonic condition or the water is in hypotonic condition, then you know that water contains low salt or higher salt. So that's how you need to know what is hypotonic and what is hypertonic. So if you talk about animal cell, if you put an animal cell in a hypotonic condition, what would happen in hypotonic condition? You know that inside the cell there is less salt and there is more water. In hypotonic condition, what is, like, if the solute concentration is less, then that means solvent will be higher, right? If the salt is less in quantity, then the water will be more in quantity. In hypotonic condition, the water will enter to the cell. That's why the cell will swell and it will cause lysis, or you can say cytolysis. In the hypotonic condition, the water will be removed from the cell, it will shrivel. And if you talk about isotonic, then the water will go inside and outside of the cell, it will be in equilibrium, or it is a perfect state. If we talk about plant cell, then the plant cell will be turgid in hypotonic condition and it will be plasmolysed in hypotonic condition. What does plasmolysed turgid means? Turgid means the process in which cell is really tight and it is filled with water, and plasmolysed means in which the condition the cell has become really small and it is shrinked and the water has been removed from the cell. And what does flaccid mean? Flaccid means that the cell is in a normal or equilibrium condition and the water is going in and outside of the cell, and it is really a good position to be in. Like, the cell wants to be in isotonic condition, not in hypotonic or not in hypertonic. It always want to be in isotonic condition. So this was about osmosis and tonicity. It is a really important concept. please try to learn this process, and don't make any mistakes by these concepts in your IMAT exam, because these concepts have been asked many times in your past IMAT past papers. So there are really high chances that this kind of concept can be repeated. Let's talk about our last topic, that is about cell wall. So cell wall is the outermost covering in plant cells. It is not present in animal cells. Cell wall is only present in plants. So cell wall is also present in fungi and bacteria, but cell wall is absent in animals. So you need to know that the animals have the outer covering only of cell membrane, and it does not contain any cell wall around it. If we talk about plants, then the cell wall of plants is made up of cellulose. The plant cell wall is made up of cellulose. If we talk about fungi, then the cell wall of fungi is made up of chitin, like this. If we talk about bacteria, then the cell wall of bacteria is made up of peptidoglycan. It is also called as murine. It is another name for peptidoglycan. So don't be confused when you see these kind of names. So murine or peptidoglycan is the same name of cell wall. So cell wall contains actually 3 layers. The first is middle lamella, primary wall, and secondary wall. If we talk about these layers, then these layers are really important because they are really confusing for so many students. If we talk about the middle layer that is present, as you can see, this is cell A and then you have cell B, and in the center of these two cells, this is middle lamella. So middle lamella is actually in the center of cell A and cell B. If we talk about the secondary layer, that is in the blue color in front of you, it is primary wall. What is it called? It is primary wall. Primary cell wall is in front of you, and the other layer that you can see in the inside is actually the secondary cell wall. So these are the 3 layers of cell wall that you can see in front of you. The middlemost is the middle lamella, then you have primary cell wall that is inside the middle lamella, and inside the primary cell wall is actually the secondary cell wall. If you want me to show the diagrams in front of you, then it would be like this. If it is a plant cell, then it is secondary cell wall, then outside of it would be primary cell wall like this, and outside the primary cell wall you will have middle lamella. So these are the layers of cell wall that is right in front of you. And if you are thinking where is the cell membrane, so the cell membrane is actually in the innermost layer. Like if you can see here, this origin line is showing you the cell membrane. So the cell membrane is in the innermost layer. So inside the cell membrane, then outside is cell wall. And if you can see here, these layer has actually a different kind of molecules that are present in them. If we talk about primary cell wall, then primary cell wall is made up of 3 kind of molecules. The first is cellulose, and then you have hemicellulose, and then at the end you will have pectin. So these are the 3 molecules that makes up primary cell wall. If we talk about middle lamella is actually made up of like gel-like molecules that contains magnesium and calcium salts. So these are the molecules that are present in middle lamella, and it also contains a small amount of pectin. If we talk about secondary cell wall, so secondary cell wall is made up of most kind of molecules. It is made up of lignin, it is made up of cutin, and then it is also made up of waxes, and you can say inorganic salts. So these are the molecules that are present in secondary cell wall. Inorganic salts. The one thing that you need to remember is that in secondary cell wall, the molecules that are present in cell wall are also present here. So the molecules of primary cell wall are also present in secondary cell wall. So that's why you can say cellulose, hemicellulose, and pectin are also present in secondary cell wall. So how does it work and how does they form? If you talk about how they are formed, then the first layer that is formed is middle lamella, and then primary cell wall is formed. Primary cell wall, you can say, is the true wall, and it is formed next, and then secondary cell wall is formed. Secondary cell wall is formed from the primary cell wall. That's why it contains molecules from the primary cell wall. So these were the layers of cell walls. The first is middle lamella, then the inner layer is primary cell wall, and the innermost layer is secondary cell wall. And then inside the cell wall is actually plasma membrane. If we talk about the difference between cell wall and cell membrane, then this is the really great table to understand this topic. If we talk about composition, then it is mostly made, cell wall is mostly made up of cellulose, and cell membrane is made up of phospholipid bilayer and proteins and carbohydrates. And if we talk about the function of cell wall is that it is really hard. Why is it hard? Because it contains cellulose, pectin and hemicellulose. If you can see here, there is lignin and cutin. So lignin and cutin actually works like this. If you can see here, these are the molecules in front of you, and lignin molecules are embedded inside these cutin molecules, and that's how the structure of cell wall is really hard, because they have cemented the lignin and cutin with each other, and this kind of bond or structure cannot be broken down really easily, by like it is really hard to break down. That's why cell wall is so rigid, so hard, and that's why cell wall gives the shape, structure and support and protection to the cell, while cell membrane regulates the movement of substances, and we discussed about all the functions of cell membrane just recently. If we talk about permeability, then cell wall does not stop anyone. It is like anyone can go through it, because cell wall contains pores and the pores are open to everyone, while cell membrane is not open to everyone. It is selectively permeable, it is semi-permeable. It only allows specific molecules to pass through. If we talk about presence, then cell wall is present in plants, fungi, bacteria and some protists, while cell membrane is present in all kind of cells. In every cell, cell membrane is present. If we talk about thickness, then cell wall is thicker, while cell membrane is a bit thin compared to cell wall. If we talk about the physical state, then cell wall is really rigid, while cell membrane is fluid, and the fluidity is controlled by the cholesterol molecules. And if we talk about the repair and regrowth, then cell wall can be synthesized, it can be deposited in layers, and it can be regenerated, while cell membrane can also be repaired through the movement and its assembly of the components. So they both can be repaired. And if we talk about electrical conductivity, so cell wall is like non-conductor, it does not conduct anything, and cell membrane can help in conductivity. That's why nerve impulse can pass through because of cell membrane. We just discussed that cell membrane helps in the conduction of nerve impulses. So this was about the difference between cell wall and cell membrane. If we write down some things that are specific concepts, if we talk about a plant cell like this, what does plant cell contains? Plant cell contains cell wall, and inside the cell wall is cell membrane. If we removed cell wall from plant cell, then what will you get? You will get an animal cell like structure, right? Then it will become equal to animal cell if we see from the outside, not in actual reality, because plant cell and animal cells have so many differences. But if you see the general picture, then removing plant cell wall will be equal to animal cell. If we removed, for example, if you have an animal cell, and if you removed cell membrane or plasma membrane from animal cell, then what will you get? You will get protoplasm. The remaining part is called protoplasm. So protoplasm is essentially two things, cytoplasm and nucleus. And let's go further. If you get protoplasm and you removed nucleus from the protoplasm, what will you get? You will get the last thing, that is cytoplasm. If you want to go further here, and let's go further. So as you can see that cytoplasm contains two parts, cytosol and inclusions. So inclusions is like the particles that are molecules that are present in cytoplasm. So if you remove inclusions from cytoplasm, like remove all the inclusions, all the hard molecules, all the molecules that are present in cytoplasm, the remaining part would be the cytosol. So cytosol would be the last thing that would be remained in the cytoplasm. And what is cytosol? Cytosol actually contains two types. What are the two types of cytosol? The first type is true solution, and the other is colloidal solution. The one is true solution and the other is colloidal solution. So these are the two types of cytosol, contains two parts. First is the true and the other part is colloidal. What is true and what is colloidal? True contains actually small molecules or ions. That is the part of true solution. Small molecules and ions are present in true solution. It is actually 90% water, H2O, and 10% is the other substances. If we talk about colloidal solution, it actually contains large molecules. So colloidal solution contains large molecules. Colloidal solution could either be sol or it could be gel. So these are the two types of colloidal solution. It could be either sol or gel. So sol or gel are really important. As you can see, like, what is gel? Gel is actually viscous or jelly like, right? While sol is not viscous, it is non-viscous. It is not like gel like structure. While sol is actually in the central part of the cell. Central region of cell is sol. And what is gel? Gel is present in the corners of the cell, or you can say in the peripheral area. The peripheral area of cell is the corners of the cell, and corners of the cell are gel like. Though you can say, like, if it is the cell, if you draw cell here, so the corners or the peripheral area of the cell is just like gel. It is viscous, it is jelly like. And if you talk about, this is the central region, it is the sol portion. It is not non-viscous and it is not gel like structure at all. So these were the two parts of colloidal solution that we discussed here. So these were the concepts that were important, and I thought that you need to know about cytoplasm, about plant cells, and about animal cells. So that's how the whole concepts are like complete here. You don't need to know anything else. I think there is one thing that you need to know about cell wall. Last thing that I promise, this is the last thing that we'll discuss. Okay, so the cell wall, if we talk about cell wall, then the sequence could be like this. As you can see, what is the sequence of the cell wall? The sequence here, as you can see, is the outermost layer is middle lamella, right? Middle lamella is present most outside of the cell. Then inside the middle lamella is primary wall, right? And then inside the primary wall is secondary wall, and inside the secondary wall is what is inside? It is plasma membrane. But what about development? So what do you mean by development? This sequence was for arrangement that the cell contains. This was the arrangement that is already present in front of you. But what if I say which layer is formed first? Like, in all the layers in front of you, they won't form first. Like, if middle lamella is present outside, that does not mean that middle lamella is the first layer that is formed in the cell. So actually the first layer that is formed in a cell is actually plasma membrane, because plasma membrane is the first layer that is formed in the cell. So in development, plasma membrane is formed first, and then after plasma membrane, the middle lamella will form. Like, middle lamella will develop. If it is a cell like this, then the plasma membrane will form, and then middle lamella will form, and then primary will form, and then secondary will form. So that's how a cell develops in actual reality. Like, if a cell is being formed from a very beginning, from the start, a cell just divided, then the first layer that is formed is plasma membrane, then middle lamella will be formed, and then primary cell wall will be formed, and then secondary cell wall will be formed. So this was the development order, but the more important order is actually arrangement order. As you can see in the diagram, the middle lamella is present outside, and then inside middle lamella is primary wall, and inside primary is secondary, and inside secondary cell wall is primary, plasma membrane. So that was the arrangement, and development order. So these both orders actually really confusing so many students, so I thought that maybe I shouldn't be leaving these kind of MCQs on the table. So if we talk about the summary of the chapter, the summary of the chapter is that there are five type of transport that are in front of you. We discussed all of them, and then the table between active and passive is in front of you. Then we studied about tonicity effect, and then the last was about membrane proteins. So these are the types of proteins that are present in your cell membrane. So this is the end of the chapter, and we discussed every single thing that you need to know for cell membrane and all the molecules and all the questions that could appear in your IMAT exam. There is no other concept that could be asked. Now after discussing this chapter, you are fully ready for your IMAT exam for this chapter. You don't need to study any other book, any other concept after you have discussed the biology from my lectures. If you are studying biology from my lectures, then that means you are already ahead of all the students that are out there. So this was all about the cell structure and function. I hope you are practicing all these questions and chapter in your practice books, in your past papers from your mock exams, and you are doing weekly full-length papers, and I really appreciate it because students are really hardworking. Students are taking lectures, but the problem is that many students don't practice these questions. Practicing questions is like the most important step in your IMAT preparation. If you are not practicing enough, then it is like a really downside for you, because taking lecture is important, but practicing is more important than taking the lectures, because now you, after practicing, you know that what is the thing that is your weaker portion, what is the concepts that you don't know, what are the things that you are recalling. So taking lecture is important, yes, but revising the concepts and practicing in is also really important. So after taking this lecture, try to solve as many MCQs as you can from these topics. Even if you are getting these MCQs from different resources, then you can practice them, but prefer IMAT Sensei practice material, because IMAT Sensei practice material is really highly relevant for your IMAT exam, and we are trying to make these kind of MCQs that will come in the real IMAT exam, right? So this was all about from my side. That was all. Bye bye, take care.